Thank you.
Hey, everyone. All right. So the first one was so much fun. I get to do an encore. But this time they're going to let me walk around the stage as we do it. So as soon as we get the screen up, there we go. Okay, so we're going to be using stem cells to deliver UBE3A. This is the one that I just told you about, where we make cells into little factories that are going to pump out UBE3A. So you guys know me. I'm not going to go through that part again. Disclosures, I actually have some this time. So first off, the work that I am presenting is not work that I myself have done. I'm presenting it as a representative of FAST. This amazing work that you're going to see was done at UC Davis in Joe Anderson's lab, Jill Silverman's lab, and David Siegel's lab, and all of their amazing students. This work is now being carried forward by a spin-off company from FAST called Transformatics. So let's get started here. We've had some introductions to gene therapies. There are two types of gene therapy. One of them is outside the body, ex vivo. One of them is inside the body, in vivo. Let's walk through ex vivo first. So ex vivo, we have our patients. We take some cells from them. This is usually a blood sample. When we do this, we put them in a dish and we grow them up. And then we do the gene therapy on these cells in the dish. We correct those cells. In this case, we're going to be putting UBE3A into them. And then we put those cells back into our patients, and those patients now have the corrected cells, and probably the UBE3A expression. Inside the body, we don't take the cells out of the patient. Instead, we put the gene therapy into the patient, and we correct the cells inside the patient. All right? For right now, we're going to focus on the ex vivo one for this talk. So we're going to be using cells called hematopoietic stem cells. These are basically cells that can turn into any type of blood cell in your body. They're like the college freshmen of blood cells. All right? Now this is helpful for therapy because they can do two really important things. They can act as cellular factories where we're going to make a bunch of UBE3A, and they can turn into cells that can actually get through the blood-brain barrier. And when they do that, they can then pump out the UBE3A so that the neurons around them can take up that UBE3A. So how are we going to deliver these cells into the patient? Well, first we're going to start with some localized chemotherapy. I know that sounds really scary, but it's localized, all right? So it's just making some room inside the bone marrow for us to have a place to put these new cool cells. So we're going to take our cells that are modified, that are going to be able to pump out UBE3A, and we're going to put them into the patient's bone marrow, where they're going to reside. Now when they do that, they're going to start making cells that are going to be able to cross the blood-brain barrier. And they're going to get up there, and then they're going to pump out all the UBE3A so that those neurons can take that up. Is there evidence for this strategy? Of course there's evidence for this strategy. Otherwise, I wouldn't be standing here telling you about it. All right? So back in 2013, an amazing researcher called Alessandra Biffi used lentiviral hematopoietic stem cell therapy for metachromatic leukodystrophy. What you need to know about this is that it's a brain disorder where it causes neurodegeneration, so the neurons die. What we're looking at right here is a scan of a patient's brain. It looks pretty normal. The impressive part, this individual with this disorder, 2 years later, the brain scan looks basically the same. Why is that impressive? Because without gene therapy, this is what that brain scan normally looks like. All right, that blackened area is where cells have died. Patients with this disorder, I want to draw your attention to the gray dots down here. This is their natural history for gross motor measurements, really low. Patients that were treated, you can see the blue, the green, and the red. Their gross motor scores are much higher, and they're increasing. This treatment really worked in these individuals. It was amazing. We've taken this at FAST with this type of strategy. We've called it UBE3A for UBE3A secreting cells. And what we've done is created a lentiviral vector that's going to carry a copy of human UBE3A gene in it. The important part that you see up here is, of course, the part saying UBE3A, because that's the protein we want to add in. And then we have the MNDU3. What is that? That's a promoter. That's what's going to turn that portion of UBE3A on so that those cells will express UBE3A. That's going to be important in a few slides. We take this and we put it into the lentiviral vector. This is going to get it into those cells so that they can start making UBE3A. Promising early results. This is in a mouse. I know it's black and white, very old school here. But what you want to see is all the little red arrows are pointing out cells in a wild-type animal that have UBE3A in the brain. You notice there are no red arrows for the Angelman syndrome mouse. But when you treat the Angelman syndrome mouse with this UB cell, all of a sudden there's a bunch of red arrows. That means that there is now UBE3A in this Angelman mouse's brain. And for those of you that love graphs, this is the level in the untreated animals. This is the level of UBE3A in the treated animals. All right? Really exciting. It goes up quite a lot. Now, we run tests on these animals to see if they have phenotypic recovery. One of the tests that is run is a balance beam test. We basically ask, are the mice gymnasts yet? So, neurotypical mouse goes across the beam really, really fast. Angelman mouse, much slower. They have balance issues. Some of them never cross, some of them fall off. What you want to pay attention to here is you see with the wild-type animal, they go across really quickly. In the Angelman syndrome animals that are treated, they go across at the same speed, if not better, than the wild-type animals. In the untreated animals, they either don't make it across the beam, or they cross it really, really slowly. They'd look like I would on a balance beam. All right, so that's all the physical part. How about the learning and memory part? We do a test called novel object. We basically ask the mouse, do you recognize something new, and do you explore it more? So with this, you can see our wild-type animals. This one right here. All the white dots, that's for the new object. They're spending more time looking at it. It's a brand new toy. Treated Angelman animals, they spend more time with the new toy too. In the untreated Angelman syndrome animals, they can't tell the difference between the new toy and the old toy. So they don't spend any more time with that new toy. So again, we're recovering learning and memory issues here in these mice. Very exciting. And it's not just in newborn treated mice, it was also in adult treated mice. So how is this going to work for a treatment for AS in humans? Well, we're going to take some of the cells, usually a blood sample. We're going to grow those cells up and turn them back into those hemopoietic stem cells that I told you about. We're going to put the UBE3A vector into them. So those cells are now going to be little UBE3A factories. And from here, we're going to take those little cells and infuse them back into the patient. They'll take up residence in the bone marrow, and then they'll make the cells that are able to cross that blood-brain barrier go up to the brain and start pumping out UBE3A. And this UBE3A is going to be taken up by the neuronal cells in the brain. Now, as always in science, there are challenges. Nothing ever quite goes as smoothly as you'd like it to. So as exciting as this technology is, there was a bump in the road. A couple gene therapy trials got halted because there was a cancer risk that surfaced. They did a comparison of two lentiviral gene therapies. One of them is called Skyzona, one of them is called Zynteglo. What the FDA decided upon when they looked at this and called in the experts to ask, what is causing these cancer risks? They settled on the promoter. One of the promoters was for beta globulin, and another is a modified MNDU3 promoter, which is the same promoter that we're using. Now with this promoter resolution, you notice for Zynteglo, they don't have a warning. Skyzona does. These were treating very serious diseases, so the FDA has let both go into actual patients because there are no alternatives. However, this impact for UB cell is that now we're going to have to look for a different promoter, all right? We want to put safety first, which means that where we were last year, we've had to take a step back. All right? But that's okay. There are many other promoters out there. We just have to find the right one for our treatment. And because everything is so promising, there's an amazing team that's been assembled to help look for this right promoter and to make sure that we get UBE3A into the Angelman syndrome patients as quickly and as safely as possible. And so I'd like to thank you once again for letting me come up here and share about this amazing treatment. And it's a wonderful community to be part of. And please, if you have any questions, feel free to find me. Thank you so much.